Welcome to today's lesson on prokaryotic and eukaryotic cells, where we will explore the fundamental differences and structures of these two essential types of cells. In our lesson today, we will start by defining what prokaryotic cells are. We will then learn about eukaryotic cells. Finally, we will compare and contrast the components of each cell type to highlight their unique features. So far, you have learned that cells are the basic units of all forms of life, and that their structures allow them to perform various functions. This foundational knowledge sets the stage for us to delve deeper into the specific features that distinguish prokaryotic cells from eukaryotic cells. Let's get started. Let's start by examining prokaryotic cells. Prokaryote cells are simple, single-celled organisms. The most common example is bacteria. Here's a breakdown of their main structures. First, the cell wall. This rigid outer layer provides support and protection. Second, the cell membrane, which is located just inside the cell wall. The cell membrane controls the movement of substances in and out of the cell. Third, the cytoplasm, which is a jelly-like substance where all the cellular activities occur. It contains enzymes and nutrients required for cellular processes. Fourth, the DNA. Prokaryotes lack a nucleus. Instead, they have a single looped strand of DNA that floats freely in the cytoplasm. This DNA controls the cell's activities and reproduction. As well as this, some prokaryotic cells have plasmids, which are small rings of DNA found in addition to the main strand. Plasmids often carry genes that provide added advantages, such as antibiotic resistance. Prokaryotes can have multiple plasmids or may not have any at all. Next, we have ribosomes, which are tiny structures where protein synthesis occurs. Some prokaryotic cells, specifically certain bacterial cells, also have flagella, which are hair-like appendages that protrude from the cell, which propels the cell body in liquid environments. Some prokaryotic cells also have a slime capsule, which provides protection to the cell. So this labeled diagram shows you the different organelles of prokaryotic cells. Make sure to note these down, as you may be asked to label a diagram in an exam. Now let's examine a bacterial cell. Take a moment to write down some of the organelles that we have already learned about. For example, notice the absence of a nucleus and the presence of a single looped DNA strand and a plasmid located in the cytoplasm. Now let's move on to eukaryotic cells. Eukaryotic cells are more complex and larger than prokaryotic cells. Animals, plants, fungi, and protists all have eukaryotic cells. Let's discuss the structures of eukaryotic cells by looking at the organelles found in animal and plant cells and discussing how they differ from those of prokaryotic cells. The most prominent feature of a eukaryotic cell is the nucleus. It is enclosed by a nuclear membrane and contains the cell's DNA arranged in chromosomes. The nucleus controls all cellular activities. Similar to prokaryotic cells, the cell membrane regulates the movement of substances into and out of the cell. The cytoplasm is a gel-like substance where the organelles are suspended. It facilitates cellular processes and contains enzymes and other molecules required for the cell's functioning. Known as the powerhouse of the cell, mitochondria release energy through respiration. Like prokaryotic cells, eukaryotic cells also have ribosomes, which are the sites of protein synthesis. Chloroplasts are only found in plant cells and carry out photosynthesis to convert light energy into chemical energy. The vacuole is also only found in plant cells. It is full of cell sap, which helps with the storage and disposal of substances. It also maintains turgor pressure in the plant. The cell wall is present in plant cells and some fungi, but not in animal cells. It provides extra support and protection. So here is our final labeled diagram, which shows the different organelles of plant and animal cells, which are both types of eukaryotic cells. Notice that plant cells share all the same structures as animal cells, but also possess additional features: a cell wall, a vacuole, and chloroplasts. Now that we have a good understanding of prokaryotic and eukaryotic cells, let's discuss some of the key similarities and differences between them. First, both have a cell membrane that regulates the movement of substances in and out of the cell. Both also contain cytoplasm where cellular processes occur. Both also have ribosomes to synthesize proteins. And both contain genetic material, DNA, though they are organized differently. However, there are some differences. First, we learn that eukaryotic cells are generally larger than prokaryotic cells. We also learn that eukaryotic DNA is linear and contained within a nucleus, while prokaryotic DNA is circular and free-floating in the cytoplasm. In addition, eukaryotic cells have various membrane-bound organelles, which just means organelles that have their own membrane, such as the nucleus, mitochondria, and chloroplasts in plant cells, whereas prokaryotic cells do not. Prokaryotes also sometimes have flagella, which propels them in liquid environments, as well as plasmids, which have additional DNA. Finally, we have learned that a cell wall is present in prokaryotic cells such as bacteria, as well as some eukaryotic cells such as plant cells. But animal cells, such as those in humans, do not have a cell wall. Whilst both bacteria cells and plant cells have a cell wall, the composition varies. Plant cell walls contain cellulose, whereas bacterial cell walls contain peptidoglycan. Before we conclude the lesson, let's consider a controversy that was recently settled in the field. The distinction between prokaryotic and eukaryotic cells was recognized as early as the 1920s. But it wasn't until the mid-20th century that scientists began to fully understand their evolutionary relationship. A key figure in this advancement was American biologist Lynn Margulis. Let's hear her explain her influence on biology.
In 1967, when I proposed the theory of endosymbiosis, it was met with intense skepticism. I suggested that eukaryotic cells, like those in plants and animals, evolved through symbiosis, with early prokaryotes merging to form complex cells. The evidence was clear to me. Organelles like mitochondria and chloroplasts even have their own bacterial-like DNA. Though many ridiculed the idea, it's now a cornerstone of biology, reshaping our understanding of evolution and the interconnectedness of life.
Great work. That's the end of our lesson today. We discussed the fundamental differences and structures of prokaryotic and eukaryotic cells. We began by defining what these cells are, noting that prokaryotic cells, such as bacteria, do not have a nucleus or other membrane-bound organelles. In contrast, eukaryotic cells, which make up plants, animals, fungi, and protists, have a defined nucleus and various membrane-bound organelles that allow for compartmentalization of cellular functions. We then examine the structure of prokaryotic cells, highlighting their simple organization, with key components including the cell wall, plasma membrane, cytoplasm, ribosomes, and genetic material in circular form. Additionally, some prokaryotes have specialized features like flagella for movement. We also explored the more complex structure of eukaryotic cells, identifying the presence of a nucleus, various organelles, such as mitochondria, and, in plant cells, chloroplasts and a cell wall. We compared these components to highlight how eukaryotic cells have specialized structures to accommodate their more complex functions and larger size. This comparison emphasizes how these features that have allowed eukaryotic cells to diversify and specialize, leading to the complexity of multicellular organisms.
Watch the rest of the GCSE biology course at Sophos Education.